In this segment, we will discuss about an important endocrine gland, that is thymus gland. So thymus is basically pink colored bilobed lymphoid mass, a tissue which is present on the dorsal side of heart as well as aorta. So thymus gland location is basically this pink colored bilobed, that means two lobes are present, lymphoid mass present on, that means location also we are discussing in the same point, present on the dorsal side of heart and aorta, right? And the origin is basically endodermal. It is endodermal origin, right? Location is on the dorsal side, that is at the back side of the heart as well as aorta. It is a pink colored bilobed lymphoid mass. Now what is this lymphoid mass? This is basically made up of lymphoid tissue, and these lymphoid tissues has lymphocytes, right? And lymphocytes, as we all know, they play an important role in the immune system of our body, right? Now this gland is maximally developed at the puberty age. After puberty, right, this gland degenerates. It undergo a process which is called as atracia. It is basically shrinking of the gland. That means size also decreases. The size decreases, activity of this gland also decreases, right? So this gland is maximally developed at puberty. That means when the growth takes place, this development of this gland also takes place. At puberty, it is developed maximum, and after puberty, it undergo atracia or atrophy, whatever you want to say, right? That is basically shrinkage of this gland takes place. The gland will become shrink, it will degenerate, right? It will look like a kind of small organ, the head of the organ, right? So this is what we call it as atrophy or atracia. Now histology wise, if we see this gland has two parts, that is the outer cortex as well as inner medulla. Okay, so histology wise it is divided into two zones. Histology, that is tissue structure. Histology wise it has two zones, right? This outer cortex and inner zone is what we call it as medulla, right? Now basically the cells, if we see this gland at a cellular level, the gland, that is thymus gland, has two types of cells, right? Thymocytes and Hessels cells. So cells are of two types. First, thymocytes, and the second is Hessels cells. Hessels cells are very, very important. Thymocytes are also very, very important. The thing is, thymocyte secretes the thymosin hormone. Hessels cells will not secrete any hormone, but these cells are phagocytic cells in nature. If you remember, phagocytosis is the mechanism or the process in which the foreign particle will be phagocytosized by the cells. It is engulfed and it is released out by exocytosis. So Hessels cells are phagocytic cells, and thymocytes basically produces hormone. This hormone is called as thymosin hormone, right? And Hessel cells undergo, or they are phagocytic cells. Now what are the functions of these thymosins? As we discussed earlier, that this gland plays an important role in providing the immunity. So thymosin role is also to provide the immunity of two types: cell mediated immunity as well as humoral based immunity, right? So functions of thymosins, let me label it first. Functions of thymosins: it helps in the specialization or the maturation of the T cells. Basically, we have two types of immune cells, two major type of immune cells, B lymphocytes as well as T lymphocytes, right? The B lymphocyte produced within the bone marrow, maturation also takes place within the bone marrow. T lymphocytes is different. Production of T lymphocytes takes place within the bone marrow, but specialization, maturation, functional T cell forms within the thymus gland. So that's why this gland is very, very important for providing the immune system or strengthening the immune system. So it helps in maturation of T lymphocytes. Now, when these T lymphocytes are mature, they activate B cells, that is the bone marrow cells, to produce antibodies. Right? Now, what are these antibodies? Antibodies are again a kind of immune molecule which are produced in response to any antigen. And what is antigen? Antigen is a foreign particle, any pathogen. It could be bacteria, it could be viral, it could be fungal, it could be any protozoa. Right? So, if any foreign particle is entering the human body or any host, the host body is designed in such a manner that immune system of the host will recognize that foreign particle, antigen, as a foreign molecule, and in response to that antigen, our body secretes antibodies, and these antibodies are nothing, they are proteins. They are normal proteins, or sometimes they are conjugated with the carbohydrate molecule, which we call it as glycoproteins. Right? So it activates B cells to produce antibodies. These antibodies chemically they are proteins or glycoproteins. It also promotes cell division. It also promotes cell division. Right? That means it is helping in the growth at early stages. Why early stages? Because this gland undergo atrophy, that is degeneration at the old age, that is after puberty. Now, these antibodies, when they are produced in the body, that type of immunity is basically called as humoral immunity. Right? So, two types of immunity or immune responses our body shows. Humoral based immunity, where antibodies are produced. This is the first type of immunity. The second type of immunity is cell mediated immunity. Cell mediated immunity, which is again the part of this thymus gland. Thymosin is only providing the cell mediated immunity. In the cell mediated immunity, antibodies are not produced in response to any antigen. Antigen is encountering in the host body. Host body recognizes that antigen as a foreign particle, and in response to that antigen, the host body will not synthesize antibody. But what the host body is synthesizing? It starts secreting immune chemicals or molecules which are called as cytokines. And now these cytokines will elicit immune response. Now these cytokines like leukotrienes, interferons, they will now participate in the immune system. Antibodies will not participate in the immune system or to provide immunity. That is the basic difference between these two types of immunity. Humoral based immunity, which is antibody dependent. Cell mediated immunity is antibody independent immunity. It is dependent on cytokines like interferons or leukotrienes. Right? Now we have written here that gland after puberty undergo atracia, that is shrinkage. Now this gland undergo degeneration as soon as the aging process continues. So it is poorly developed, but it is degenerated in case of adults. That's why we observe that adults, that is basically old age, till puberty it is maximum growth, maximum developed gland is there. Right? After puberty it undergo shrinkage, that is atrophy or atrophy. Now when an individual reaches up to the old age group, we found that this gland is completely degenerated. Right? A small pin is observed when we test for this gland. That is why the old age individuals suffer from a lot of infectious diseases. Why? Because the gland which is providing immunity is now no more functional. It is a very small gland. It is degenerated. So who is going to provide immunity? No. So in case of old age, the immune power, immune system weakens. That is the reason is atrophy of thymus gland. So this was all about the thymus gland. In the next segment we will discuss about the pineal